Dear all, welcome to this video. Please hit the subscribe button so that you automatically get updates for upcoming videos and lectures. Thank you. Enjoy watching.
So before we go for immunology or actual defense mechanism, we will see some terms related to immunology. First is pathogen. As I have already discussed what pathogens are. Pathos here is a Greek word which means suffering, and gen means to produce a disease. So obviously the one producing a disease or the one producing a suffering is called as pathogen. All the microbes which causes diseases are called as pathogens. Pathogenicity is the capability of causing a disease. Virulence, as we have already discussed, degree of pathogenicity or the strength of the microbe to produce a disease. Attenuation is decrease in virulence. Example is vaccine. Exaltation is increase in virulence. Antigens. Now, what are antigens? Antigens are usually substances which are produced in the microbes, usually in microbes, and when introduced into the human body, or not only in human body but in any host cell, it will result in the stimulation of production of antibodies. And these antibodies will react with these antigens will either react with these antibodies, and if it is a second exposure with the antigen, then the whatever antibodies are present inside the host cell, they will react with the antigens. Now, what are antibodies? Antibodies are the immunoglobulins which are produced by the animal cells on exposure to antigen, or rather instead of saying exposure, you can say on introduction of that antigen into the body, these antibodies are produced. Now, what is antiserum? Antiserum is the blood serum which contains antibodies. Immunity. Now, what is immunity? It is the resistance offered by the host to the harmful effects of pathogenic microbial infection. So, whatever microbial infection happens, to overcome that infection, whatever body shows resistance, either to overcome it or to avoid it. So, that is the immunity of that particular host. Susceptibility is lack of the power of body to resist the infection. That is, a particular person is not capable or is not able to overcome the pathogenic effects of the microbial infection. So in that case, you will say that that particular person's immunity is low, or you can say that that particular person is susceptible to the disease. So susceptibility term is usually applicable for whom? For either pediatric or geriatric patients, because for pediatric patients their immunity is not well developed, and in other case, that is for geriatric patients, the body has undergone wear and tear, and due to which their immunity power has been decreased. Next is immunology. It is the study of immunity, and immunological preparations are all the preparations which are used to increase the immunity. Example is Chyawanprash, which is readily available in the market. Now, what are the defense mechanisms of the host? Now, these defense mechanisms can be classified into two groups. First is the natural resistance, which is also called as constitutive or innate resistance, and the other is the internal defense mechanism. Now, this natural resistance is further classified into four classes, that is species, racial, individual, and external defense. Now, species here, it means that some diseases are caused only to humans. Some diseases are caused only to animals. Example is rabies. Rabies is not caused to humans. It is only caused to the dogs. So that becomes species, because that particular rabies is only limited to the dogs and not to any other species. Next is racial. Now here racial, it means that some white people, they are more susceptible to some diseases as compared to the black ones or the Negroes ones. So usually you must have seen the white people, they easily get infected with typhoid and all, even in minor conditions. But in case of Negroes and all, usually South African people and all, their immunity power is so high. Why? Because of the external environmental conditions, they have been adapted to that particular environment, and due to which their immunity has been increased. Next is individual. Now, though a particular person belongs to the same species, though two people are there, They both have same racial difference, no racial difference, they are in same family, but it may happen due to individual or genetic combination, due to genetic arrangement, that a particular person's immunity is high or low. And next is the external defense mechanism. We'll be discussing what harms under external defense mechanism. And on the other hand, internal defense mechanism, it includes specific and non-specific defense mechanism. We'll be discussing these things in detail. Now here you can see the external defense mechanism. Now right from the top of the body to the bottom, we will go one by one. Now lysozyme are the enzymes which are present in CSF, that is cerebrospinal fluid, helps the brain to prevent infection from the microbes. Then the mucous membrane, or the mucus which is secreted in your mouth continuously and also in your nose, it helps in trapping the microbes if any present, and this mucus also contains lytic enzymes which, if any bacteria enters, they will be killed. Then the cilia lining trachea. Our trachea is made up of cilia. They are small projections which usually entrap the microbes if inhaled by the humans. So it will prevent the microbes entering into your lungs and causing some severe infection. Then the sebaceous glands. The sebaceous glands are the glands which produces sebum or sweat. So they form an oily layer on the body, on the whole skin, and they also drop the pH of the skin. Due to the drop in pH of the skin, all the neutrophilic alkaline loving bacteria, they cannot survive on the body, and hence they get easily killed. And other thing, even the sebum glands, they also contain some enzymes. They contain high percentage of oil, so all microbes cannot sustain or they cannot live on that particular environment. Next is the acid in stomach. As you all know, the pH of the stomach is 1.2. It is made up of acids. So the all neutrophilic or alkalophilic bacteria, again, they cannot, even if they are ingested into the body, but they will be killed by the acid. Then commensal organisms in the GIT. GIT usually contains some microbes, that are lactobacillus, E. coli and all, which help in digestion process. And on the other hand, if any other bacteria is getting infected, they will also be killed. And there are some organisms, microorganisms, which are also present in your vagina. They also help the vagina to keep clean and to fight back any infection if it takes place. So this is the way how the external defense mechanism works. Next is the internal defense mechanism. Now, under this internal defense mechanism, as we have already discussed, there are two types, specific and non-specific. So today we'll be discussing non-specific defense mechanism. Now, under this non-specific defense mechanism, there are different cells which offers this: phagocytosis, natural killer cells, interferons, inflammatory response, fever, and complement system. Now, phagocytosis. Now, as you all know, this process, we will discuss it in detail. So, initially, what these phagocytes do, they usually introduce a pseudopods. Now, what this pseudo means, false, pods means heat. So, they introduce or they elongate pseudo pods, and that is the false arms, and they form a vacuole. Now this is your microbe and these two are the pseudopods, so these have been elongated and they have formed a vacuole. Now this vacuole is known as phagosome or phagocytic vesicle. Now this phagosome, it combines with lysosome. Now lysosome is a ball, you can say, or it is a vacuole containing all kinds of lytic enzymes. So when this lysosome combines with the phagosome, it is known as phagolysosome, and it will cause the breakdown of that particular bacteria due to the presence of the lytic enzymes. All the bacteria, or they will be partially digested microbes. So this is your phagolysosome. Okay, these both are your phagolysosome. This is the initial stage, this is the later stage. And last is the residual body. So whatever indigestible material is present after the process of digestion, this will be removed. That is a discharge of the waste material. Okay. So now the start of this phagocytosis takes place with the help of chemotaxis. I hope everyone knows the meaning of the term chemotaxis. Chemo means chemical and taxis means movement towards a chemical. So these pseudopods secrete some chemical due to which the bacteria get attracted or the microbe get attracted towards the phagocytes, and later they adhere to the walls. Now, during this adherence process, it requires cations, that is calcium and magnesium, and some electrostatic forces. And even opsonin fixed attachment also takes place. Opsonins are the small projections which are present on the pseudopods, okay, which helps in the attachment of that particular bacteria. Okay. Now, the killing process of the bacteria is usually divided into two parts. One is the oxygen dependent, which is also called as the respiratory burst, where different kinds of oxygen species radicals are formed, like superoxide radicals, singlet oxygen, hydrogen peroxide, hydroxyl radicals. So these radicals, they cause oxidation of that particular bacteria and result in the death of that microbes. Then the second part of killing is known as the non-oxygen dependent killing, which involves lytic enzymes, which are present in the lysosomal granules. Okay, so these lytic enzymes usually include peroxidase, catalase, ribonuclease, lipase. So these, they carry out the non-oxygen dependent killing process of the bacteria. Now, the combined action of this oxygen-dependent process is also called as the respiratory burst, and the combined action of the respiratory burst and the lysosome granules, they will cause final killing of the bacteria. So, usually gram-positive bacteria, they are easily killed. Gram-negative bacteria are persistent, and it becomes difficult to kill them. And there are some bacteria like mycobacterium which are resistant, and they can even multiply in the phagocyte. They will enter inside the phagocyte, but still they can multiply. So here you can see that the virulence factor of that particular microbe is high, as it can inhibit the phagocytosis process. So our next class is the natural killer cells. They are also known as null cells. Why they are called as null cells? Because they don't have surface antigen. So since in the they don't have any antigen present, so they can kill any kind of cells. Okay, they can kill cellular target without prior exposure to antigen. Antigen and all things are not required, usually applicable for cancer cells and virus infected cells, but the exact mechanism how they work is still not known. Next is the interferons. Now, interferons are usually the proteins which are secreted or which are produced by the eukaryotic cells on exposure to viral infections or and double-stranded RNA. So double-stranded RNA viral infection, if you get, then in the body will produce some proteins which are called as interferons. Now these interferons are further divided into three classes: alpha, beta, and gamma. They don't directly react with the virus, but what they do is they will produce some antiviral proteins which are non-specific in nature. Since they are non-specific in nature, they are applicable, means these interferons can kill all kinds of viral infection. Not all I can say, but they are being non-specific, they can open to many viral protein destruction. Okay, so now these purified interferons, they are stable at low pH and have fairly heat resistant, and they are unstable at physiological conditions, and they resist viral infections, inhibits the cell proliferation, and promote cell death. That is the cytotoxic activity of the natural killer cells. And next is internal defense mechanism is the inflammatory response. So whenever you have some injury, usually what happens is due to that injury, irritation takes place because the microbe has already entered into due to the cut or wound or any kind of sting. So once that microbe has entered, so what will happen is, due to its sting, the arterioles first will constrict. Why they will constrict? Because if there is any bleeding, if there is any cut, what the body has to do, it has to first stop bleeding. It has to stop the flow of blood. So that's why all the arterioles, they contract initially, and once the blood stops, then they dilate. Why they dilate? Because what they have to do is they have to increase the blood flow to the site of injury. Why they have to increase the blood flow to the site of injury? Because if the blood flow is increased, large number of macrophages, leukocytes, neutrophils will enter, or they will move to the site of injury, and they will cause the killing of bacteria if any present, and they will cause the healing process faster. Okay. Next is fever. Now fever is rising temperature following infection. It is a natural defense mechanism, usually due to increase in body temperature, uh, and many microbes, they cannot resist high temperatures. They easily, either they get killed or they get inhibited. Their activity is inhibited. They also stimulate production of interferons and endotoxins and interleukin 1. They usually cause fever. Now next, one of the important internal defense mechanism is the complement system. Now this complement system, it is also called as C system, which usually gets activated. It first of all consists of many proteins, okay, and they are usually activated on introduction of antigen antibody interaction. Means once an antigen antibody interaction takes place, this complement system will get activated. Now usually it consists of 11 proteins, C1 to C9. And the C1 is made up of 3 subunits, that is C1Q, C1R, C1S. So that's why the total count comes to C11. Now here you can see the different sites of synthesis, like C1 is synthesized in intestinal epithelium, C2, C4 in macrophages, C5, C8 in spleen, and liver, C3, C6 and C9. Now, what are the properties of these complement proteins? First of all, they are present in set of animals, birds, and fish. Everywhere they are present. They are non-specific serological agents. So, this complement system does not get activated only on the presence of any viral infection or bacteria. Usually, it gets activated on any kind of infection. It doesn't increase the immunization. Okay. So if your body's complement system is working well, it doesn't contribute that your immunity is good. So it doesn't increase the immunization. Doesn't bind to free antigen. If there are free antigens or antibodies present in your body, then the complement system will not get activated. There has to be interaction between antigen and antibody. Then only your complement system will get activated. They are destroyed at 56 degrees Celsius in 30 minutes. And IgM, IgG1, G2, G3, they react with the complement. Now there are two pathways of this complement system through which it works. First is the classical pathway, which contains 9 proteins, and the next is the alternate pathway, which contains 13 proteins. And very important, activation takes place only on the interaction of antigen and antibody. So we will first see the classical pathway. Now this is your microbe cell. Okay, it has been introduced into your body. Now this cell, microbial cell, will obviously have some surface antigen to which these antibodies which are already present in your body have been attached. So here it indicates that the antigens which are present on the surface and the antibodies have reacted with each other, and thus antigen-antibody reaction has already taken place. So since now antigen-antibody reaction has taken place, this C1 protein, which is made up of C1Q 1 subunit, C1R 2 subunits, and C1S 2 subunits. So this will get, first of all, the C1Q portion will first of all link to the Fc region of the antibodies, and the C1 will get activated. Once this C1 subunit is activated, the C1 will get converted into C2 and C4. Okay, it will activate. It doesn't get converted. Actually, it activates C2 and C4. Now, further, the C2 will get activated into C2A and C2B, and C4 will get activated into C4B and C4A. Now, this C2B and C4B, that is known as C3 pro-convertase. So, this will activate the C3. So, that's why it is called as pro-convertase. It will activate C3 subunit, and this C3 subunit will get converted into again C3A and C3. One more thing is important, which I would like to bring into your notice. All the A portions which are present, those are all smaller subunits, and the B portions which are present, that is the one containing the B subscript, are the larger subunits. Okay, so now this C3A and C3B further, now this C3B will further combine with C2B and C4B, and they will together form C5 convertase. Means the C5 convertase usually contains C3A, C3B, C2B and C4B. And this the C5 convertase will activate C5, C5 will activate C6, C6 will activate C7 and C7 will activate C8 and C8 will activate C9. So once all these channels have been activated, they all will form a transmembrane channel, which is also called as membrane attack complex. Now this membrane attack complex, it is a cylindrical portion, and it is cylindrical in shape, and it will cause leakage into the cell contents, leading to cytolysis. So here is the summarization of the complement system, where you can see on the left side here it is a classical pathway, on the right side it is alternative pathway. Now this alternative pathway, here you can see the C1, C2, C4 are involved. Here it is C3B, factor B and factor D is involved. We haven't yet discussed it, I'll discuss it later. So they will all activate the C3, C3 will get converted into C3A and C3B. C3B portion in the presence of magnesium ions will cause opsonization, and the C3B will activate C5, C5 will get converted to C5A, and this C3A, C5A will be responsible for inflammation, chemotaxis, release of histamine and serotonin. All the inflammatory responses will get activated with the help of C3A and C5A. Now this C5 will get converted into C5B. B is C5A and C5B, and out of which this C5B will activate C6, C7, C8 and C9. And further it will lead to formation of MAC. MAC is the membrane attack complex, leading to cell lysis. Now this is the second pathway by which the complement system gets activated, that is the alternative or properdin pathway, where the antigen antibody complex is not required. Here what will happen is directly the cell wall polysaccharide will combine with C3, then this C3 will get converted into C3A and C3B. This C3B will combine with factor B, and in the presence of magnesium ions it will form C3BB. This C3BB is exposed to factor D, and which will lead to formation of C3BB and C3BA, and this C3BB forms the C3 convertase, and it further the same classical pathway is followed. So in the initial stage here what we saw, that further all this leads to the activation of C3 and the same pathway then continues. Now what complement proteins are responsible for what kind of activity? So C1 to C9 all are responsible for lysis of virus infected cells, tumor cells, protozoa and bacteria. C1 to C5 are responsible for endotoxin activation. C1, C2, C3, C4 are responsible for virus neutralization. C3b is responsible for anaphylatoxin release, opsonization, enhancement of cell-mediated toxicity, stimulation of production of beta cell lymphocytes. C3a and C3b are responsible for the enhanced induction of antibody formation. So antibody formation is improved. And C5a is responsible for chemotaxis of the neutrophils, eosinophils, and monocytes. Now, what are the biological effects of the complement system? It is responsible for bacteriolysis and cytolysis, so cell death and bacteria death. It is released during the cascade reaction. It amplifies the inflammatory response, because we have seen that C3 and C5A are also responsible for the inflammation, or inflammatory response are amplified. This complement system, it also participates in type 2 and type 3 hypersensitivity reactions. In any autoimmune disease, the amount or the concentration of this complement system lowers down. Then C3C6 it also helps in coagulation purpose, and C bound to antigen antibody complex adhere to erythrocyte. We will stop here. The specific immunity we will discuss later. Thank you.